Hello and welcome to the April Snomed Ct research webinar. I'm Susie Roy, the host of the research webinar and from Snomed International, and thank you for joining us today. I know after a year of doing this, everyone is very used to Zoom webinars, but just to go over a little bit of housekeeping, you'll notice that everyone is currently muted. If you have any questions as we go along, please go ahead and enter your questions into the Q&A box, and we'll have a Q&A portion at the end of today's presentation. As always, this meeting is being recorded, but both the video and the slide deck will be available later this week. As many of you know, this webinar is part of the Snomed Ct web series, which have become immensely popular events. In addition to this research webinar, we also have a clinical track and an implementation track. These other webinars focus a little bit more on clinical implementation and use of Snomed Ct versus our research webinar, where we focus on any research on or with Snomed. As always, all of the webinars are free of charge. You just need to register to attend live, or of course you can watch the webinar recordings later. You can visit snomed.org/web-series to see upcoming presentations for any of the webinar tracks. And of course, if you missed any of our webinars over the last year or so, you can view those or any other videos on our Snomed International YouTube channel. To find that, you just go to YouTube and search for Snomed International, and you'll find all kinds of videos from our organization that I highly recommend. And of course, I still have our research reference group going. So if you'd like to be notified of upcoming research related news and events, you can join our reference group. You just need to email me, sro@snomed.org, to join, and I will get you on that list. We usually have about 3 emails for the month, so it's not going to clog up your inbox, I promise. And I have one last little plug to announce for today before we go on to the main event. Our call for abstracts for our annual Snomed Ct Expo has been extended. So for those of you who are new to the Snomed community, first, welcome. So our Snomed Ct Expo happens every October, and that is where you, our Snomed community, showcase your current work with Snomed Ct. And so our submission deadline has just been extended to May 7th, and I highly encourage all of you to attend. Last year, our virtual Expo had almost 2,000 people from 60 different countries around the world joining. So I highly, highly recommend, if you wanted to have a nice wide audience for your Snomed work, to think about submitting. If you have any questions about the submission process or even just about the Expo itself, please don't hesitate to contact me. Again, sro@snomed.org. Okay, and so now for the main event. Dr. Rachel Richardson is a professor in the Department of Learning Health Sciences, School of Medicine at the University of Michigan in the US. Dr. Richesson conducts original research on the quality and usability of data from electronic health records. She has directed implementation of data standards for a number of multinational, multi-site clinical research and epidemiological studies, including the NIH Rare Diseases Clinical Research Network, Type 1 Diabetes TrialNet, and the Environmental Determinants of Diabetes in the Young, the TEDdy study, and the National Distributed Patient Center Outcomes Research Network, or PCORNet. Dr. Richesson currently co-leads the EHR core for the NIH Health Systems Research Collaboratory, which is developing approaches for using EHR systems and data in programmatic clinical trials. In addition, she and Charles Friedman co-lead the multi-stakeholder mobilizing computable biomedical knowledge community, establishing the standards, policies, and governance needed for biomedical knowledge to be widely disseminated and applied. Today, Rachel will be presenting Using Snomed Ct Relationships for Data Exploration and Discovery in Rare Diseases, an example in Urea Cycle Disorders. And with that, Rachel, I'll send it over to you.
Okay, terrific. Thanks so much, Susie. And let's see, let me just make sure everyone can hear me and see my slides okay?
Yes.
Okay, perfect. All right, well, thank you so much. And it's really great to be here and see some familiar faces in the participant list, including a few folks that have worked on this project. So I encourage them to participate in the question, answer, and discussion section when we finish. I'm going to do my best to do justice to this great work that's been going on for off and on more than a decade since I started with the Rare Diseases Clinical Research Network in like 2004 actually. But we've really had some breakthroughs by bringing in some really key people and key thinkers at different points in this. So it's really exciting for me to share, and I'm going to try and tell this just sort of a bit as a story of how our collaboration evolved. So first I'll say I don't have any conflicts of interest or financial relationships to report. And then before I really get into sort of a description of a data visualization tool in this story of how we use Snomed in one of our rare disease research network studies, I normally give a little Snomed background, but it's terrific. I don't have to do that for this group, but I'll tell a little bit about the history of our collaboration and our data set and what we're shooting for. And then I'll really end with sort of the challenges and a lot of directions we want to go for next steps. And I really hope that we can have a good discussion, and I'm really looking forward to people's reactions and comments and suggestions about how to move this forward. This is really just the tip of the iceberg of I think what we could do with Snomed. So when I started this, I worked for the Data Coordinating Center for the Rare Diseases Clinical Research Network when it began in 2004 at University of South Florida. And we were in a position to coordinate the data entry and work with study design and the analysis of all the studies in the network. And so there were a number of clinical trials, but there were definitely a large number of natural history studies. This was really the emphasis for these diseases that were rare and sort of by definition under-researched. And so I always saw the potential of Snomed and all the semantics embedded in it as something that would really lead us to some sort of discovery, just some eureka aha moment about maybe we could cure, or identify treatments for, a dozen different rare diseases at once. That's really how I sort of went into this when I saw Snomed. But I also saw Snomed as really the terminology that was going to underlie electronic health records. And even though the research network, the Rare Diseases Network we were doing de novo collection, we were collecting data specifically for these research studies, I always had this eye that I wanted to connect with what was going on in the clinical world. And so Snomed CT was always appealing to me right from the beginning. And again, I was just taken with the semantics of Snomed CT, the concept base, the idea of lots of descriptors, and particularly the hierarchies. And this idea that people could code at all different levels, and some people could get very, very detailed in how they coded things, and others could be more broad or even a little more superficial, but at the end of the day, we'd have this ability to roll things up. And then I saw these attribute relationships as something that would really lead us to this discover, we're going to cure a dozen rare diseases in my first few years with the research network. So I had lots of high expectations for Snomed CT. And then when I started with the Rare Disease Network, really my task was, embed standards and data standards however I could and wherever I could in the network. Just for background, the Rare Diseases Network now is got 23 different research consortia. Each is a partnership of multiple sites, multiple investigators, support with patient advocacy groups to identify questions and conduct research in diseases in certain areas. So there's the Rare Lung Disease Consortium, the Rare Liver Disease Consortium, a number of consortiums that really in diseases, over 150 diseases were studied in the network, really nothing in common in terms of etiology or pathology, except for the fact that they were rare. And so I was really challenged about how to implement standards in this context. But one of the things that I did that I was real excited about was to be able to embed these Snomed browsers and get some Snomed coding as part of the data collection for all these studies. So again, this was 2004, 5, or 6 or something when we started these first studies. So this interface isn't wonderful, but here's an example of the kind of forms that we built at our data coordinating center. This is a history form. There's structured variables on there where research nurses and research physicians that were conducting clinical visits could document what they saw, but there were also places to put other findings, as you can see. And so we built an interface where folks would type these findings, and then they would hit a button, and they would be populated into the form and hence into our database. We had these in our physical exam forms and our medical history forms. So here's a medical history form that's organized by body system, skin, ear, nose, throat, et cetera, or different categories. And again, users could enter their findings, hit code. They would be taken to, really we connected to a terminology server hosted by Apollon. And then they could navigate up and down the hierarchies and choose codes. And then they would be populated into the forms and hence into our database. And so these were used in a distributed way at the different research sites for each of the studies. And so let's see, yeah, so this is added to the form. And there was no limit to the number of SNOMED observations that people could enter in these data sets. So there's potential here of having lots of observations in rare diseases in these curated data sets as part of these studies that were prospective and had very prescribed observation and follow-up periods there. And so this just continued to grow, and I knew there was potential, but it took us a while to get there. One group I want to highlight in particular, and this is really the focus of this collaboration in this talk, is one of the rare disease consortium called the Urea Cycle Disorders Consortium. And this is still run out of DC Children's National Health Center, and Mark Batshaw was the PI at the time, and now it's Andrea Gropman from DC Children's and Sandesh Nagamani from Baylor College of Medicine are the co-principal investigators of this. Again, this is a consortium of investigators, I think a dozen different sites, number of different studies, but their showcase was really a natural history study across the urea cycle disorders, a set of diseases. And again, I saw for this group did really well on the SNOMED coding. I mean, they embraced it more than any other group. You can imagine, you saw those forms are, it's at the end of the day, it's documentation. We're requiring people to search and browse and enter terms. And so different studies sort of had different levels of enthusiasm for it. But the Urea Cycle Disorders Consortium really did a great job of embedding these this coding into their monitoring, it working with us at the data coordination. We're coding, checking codes, finding missing codes, that SNOMED people navigate and that kind of thing. So they really, in my mind, of all the maybe 24 natural history studies in the network, really had the richest and most complete SNOMED data. And so it took a number of years, of course. You're chugging along on these studies that go on year after year before we're in a position to really use this data. And what we had available to us at the time was just sort of the SNOMED tables and really the relational database format of the SNOMED concepts and relationships. And so it was really a challenge for us, and for me in particular, not having a lot of technical skills and not having the folks, even the folks that were interested in the data were looking at some of those structured variables a little bit more than the SNOMED variables. So to try and sort of put this process combine that with our database and do some discovery was just really a challenge. And I remember at the time sort of dealing with the issue of transitive closure. And so I had this vision of our data is just, we're gonna code things, everyone's gonna code in SNOMED, and we're gonna look at the data and things are just gonna cluster in some way. We're gonna find, these patterns that are really important or significant, or even just interesting in this condition to sort of give us hypotheses to move forward. And there were just a lot of challenges with that, just in terms of getting our head around it and then getting the motivation to do it. So we didn't do really anything with looking at these SNOMED data at the time, the maybe 6 years that I was with the rare disease clinical research network. Studies are busy, life goes on, and a lot of detours happened over the years, but I kept touch with the investigators from the Urea Cycle Disorders network, in particular Bob McCarter, who's an epidemiologist who functioned really as the statistician for all these studies. And Bob was always interested in Snomed Ct and how can we use it. And so every couple of years we would talk and we'd sort of delve into the data. We'd run some frequencies and kind of see what can we get out of this? Where's our discovery? And it was always just sort of a little bit more work than we could manage. And we just never got too far, but just sort of kept touch. Then finally, years later, job changes happened. I went to Duke University and later to University of Michigan. But Bob reached out and sort of timing worked when I was at Duke, that there was a student named Marcy Bowen, a nurse and a nurse informatics student, and now a stellar informaticist and data scientist, actually, who was really interested in Snomed Ct. She had taken my data standards course and wanted to do a project, really a volunteer project, looking at data. And so the timing just worked out that Bob had called and said, let's start looking at some of this data again. People are asking me questions about some neurological and behavioral findings, and maybe that's a way we could subset this and look at this. So I brought in Marcy, and at that time, the natural history study, as you can see on the left, it had accrued 733 patients that had 10 years of follow-up. And these are twice-a-year visits with all kinds of cognitive testing, samples, blood samples, genetic testing, genetic confirmation of disease, and then really detailed physical exams and medical histories at all these different time points. So it was just a really rich data set. And we wanted to really explore these neurological and behavioral findings. As we dug in there, we found that there was some missing data and some dirty data, or some things where people had not coded. They entered the text, they sort of didn't code, or they did things that they used a navigational term in SNOMED and that type of thing. So Marcy came in and really spent a lot of time cleaning this data set and working with it, validating some of the SNOMED codes there, adding codes where the text was there, but they didn't enter the code, and really enriched it for us and started to look at it. Which was just great. She moved us forward in a way. She did all kinds of data, frequencies, and said, okay, even we've got, obviously we're seeing lots of findings and clinical findings and disorders in our data, but also some findings and procedures and observable entities creeped in as well. We could find different ways to query it. So we could sort of tap into transitive closure tables and start to say, well, how many different seizures and things do we have? But Marcy really she honed in on what I suspected, but she really brought some visualization to the fact that we actually had a lot of really granular data that needed to be rolled up, if you will, aggregated into like ways. And so she found that most SNOMED codes occurred with only one patient. So here the very small boxes, or the box size correlates to the number of patients with this code. And she found that the great majority of the codes, the unique codes that were in our data set, occurred in frequencies of 25 or less. So this again is just sort of, it was just more incentive of how can we roll this data up in ways that, how can we even help investigators understand it or determine patterns. And it would be one thing if we could say, well, let's look at all the seizures, let's look at the pain, let's look at headaches. So we kept coming up with what are conditions of interest, and then you could still see that these codes are sort of becoming rolled up into useful ways, but we really had this vision that the data itself would just somehow speak to us. And so Marcy explored all kinds of different visualizations and started moving towards looking at graph representations. And she tried all kinds of off-the-shelf products and reached out to all kinds of data visualization experts at Duke that I'm very grateful for assisting us. And we did some posters on just sort of the nature of our data set, the nature of the SNOMED codes and all the potentials there. But at the end of the day, we really had these hairball kind of things, right? These visualizations that really didn't tell us what we thought. And we were just it was frustrating. And at one point we even took the Snomed codes and mapped them to ICD-10 codes and just used the highest level classifications there as a way of just sort of sharing with investigators the kinds of things we had, but we knew we could do better. And then Marcy stumbled on this great work from University of Nebraska. And I think Scott is joined today. I don't know if Jay has, but Scott Campbell and his team used a graph database version of Snomed Ct. They put Snomed Ct into a graph database, a Neo4j database, and demonstrated the effectiveness of the way that they could do queries in just a much more efficient manner, a much more direct manner. And it was really just incredibly enlightening for us. And Marcy reached out to the team, and they were just wonderfully receptive and collaborative about a partnership. So then we started working with Scott and Jay and leveraging their great work in this area. And this is just a screenshot from their paper in Journal of Biomedical Informatics. I should have put this source here. I'll put that in for the notes when we distribute this. But basically a graph database version, I don't know how familiar people are with it. In contrast to the tables of a relational database, this is really, we've got your concepts here as nodes. You have nodes and edges basically, and you're not unlimited into how many connections you could make here. So in the center, we have Snomed code pneumonia disorder as a center node in purple. And then the relationships are these edges. So we see the ISA relationships above to lung consolidation and pneumonitis. And if you recall earlier on the transitive closure slide, these kinds of things could be sort of pre-calculated, if you will, or pre-populated into tables for querying. But here you could just throw out, you're really unlimited in a graph database as to how many relationships you can have and how many relationships along a path you can have. The descriptors are also here as nodes on the left, or the synonyms, and then that relationship that it has description is an arc or an edge on the left. And then you can have all the different semantics of Snomed Ct in this form, where you've got the concepts as nodes and the relationships as edges or arcs. And so the challenge then became, well, wow, how can we leverage this? So we had all of Snomed, all the knowledge that's known. So to me, this was just the greatest knowledge base, that I could imagine to combine with our data. So we would take part of our Urea Cycle Disorders data from this natural history study, 733 kids, lots and lots and lots of visits. So Marcy did a lot of work to just make sure that we just pulled out just the Snomed observations that were associated with a baseline visit. And then we wanted just one observation per person. If for some reasons somebody had entered the same code at multiple points on a form, which does happen. But we moved we basically took Neo4j, took the Snomed Ct version that was in Neo4j that Scott Campbell and Jay Peterson from University of Nebraska have shared. And then we added our data tying the participants to visit. So the study participant to a visit. And in this case, it was just baseline, just to simplify it. And then the baseline visit had many, many, many observations. And then that's how we started. And we really limited the variables, but later I'll tell you, we did bring a few more in. So then we got something like this. And we still were just like, how do we find the pattern? We had so much, so many relationships there, we just couldn't even see anything. And part of our challenge that took us a real long time was just to figure out how to filter through, how to get only the Snomed codes that were related, that were in our data set, and only the relationships that were attached to those codes. So it actually took us some time to filter through these and begin to make sense of it. And this one slide really represents like 18 months of Marcy kind of running around and trying different things, and lots of collaborators trying to help us on how can we visualize this. And it still came down to people saying, well, what do you want to know exactly? What are you searching for? And then we'll figure out how to do the queries and get what we need. But we really were just trying to discover stuff, and it was very, very loosely defined. So that was our challenge, and we were stuck for a while. And then a couple of things happened that really brought us to what I think is a breakthrough novel. share with you now our story. And it happened really quick once we got these two pieces. One was we got a tool that was developed by Tom Beaumont, who's actually joining us today. He's a data scientist from Duke, Office of Research Computing, and he came into this as a curiosity and with a helpful attitude, but unfunded, and helped us build this tool that allowed us to start querying interactively with the data and combining variables in a way that we hadn't thought about. And then the second thing is we brought in a deep domain expert, which I thought we had domain experts on our team. We definitely had folks that were active in urea cycle disorder research, very active in the studies, data analysts, true, but they were really had been embedded in this research team for years and years. And so, and familiar with the questions and designing the studies and on protocols and running the analyses and things. So we thought we had experts in, but it wasn't until we really brought in somebody with the introduction of a tool that could then start looking at the data and generate questions. And then, so I'll just sort of quickly then give you a sense of what, you know, the breakthrough we had, and just give you a sense of the visualization of this. And then I'll talk a little bit about sort of our limitations and challenges and next steps, and then open it up for discussion. But just as background, what our discovery came around the idea of facilitating some inquiry and hypotheses that a scientist working with urea cycle disorders, very specifically with doing a lot of work with animal models, had some hypotheses and came to Bob McCarter and the UCD team to really look at this data to sort of confirm and identify some other lines of inquiry around this. And so the distinction, or the really important issue, just to know as background, is there's actually 8 different types of urea cycle disorders, and they all are the result of some sort of defect in this urea cycle process, which this is the urea cycle is the mechanism for clearing waste nitrogen resulting from protein metabolism. So, and if the urea cycle doesn't function properly, then you have a buildup of ammonia called hyperammonemia. And there's all kinds of, that's a neurotoxin and a toxin to the body, and it can cause all kinds of symptoms, very severe neurological symptoms, and can be fatal. And so that is a challenge. It's a phenotype that all these disorders share, but the disorders themselves have a different mechanism. They all result from a deficiency in this urea cycle disorder, but of different parts of it. So on the right, I've got just, these are the 8 disorders, and I'll spare everybody my pronunciation of them. But each one is a deficiency of a certain enzyme or a precursor to an enzyme in this cycle. And they all have some sort of sequelae related to hyperammonemia, but they also have different phenotypes. And so this investigator, Sandesh Nagamani, wanted to sort of distinguish between these different subtypes and try and find a mechanism of action that was different and, you know, potentially a drug target there. So the story, so now that you sort of have a sense that there's different subtypes that have different enzyme deficiencies, similar phenotype, but Sandesh came into this looking at our tool, which I'll show you in a minute, but with this sort of background. He was really looking at some urea cycle disorders that are expressed in multiple tissues outside the liver. This has been known, and that these disorders then should present symptoms even if they don't have this hyperammonemia. So as I mentioned, the excess ammonia is toxic. That can cause all kinds of neurological problems. So it's not uncommon to see neurological deficit or developmental delay in in children and adults that have these conditions, but you would expect that as sort of, that's the sequelae from this hyperammonemia. The question was, does this disease manifest in different ways? Are there other mechanisms happening with the diseases, or other pathology that's happening sort of in other body tissues, in particular in the nervous system? So Sandesh wanted to explore the phenotype data from these, looking at the different disorders to really drill down into certain neurological symptoms, and then also this combination of their disease severity, if you will, or their hyperammonemia there. So to do this, before we could even get started, he couldn't even, or maybe he probably could have articulated his queries. We didn't bring him in early to decide that, but the story I'm going to tell, I think, evolved from him looking at the data, and that's really the point that I wanted to make. There was no way, we weren't ready, even with this sort of hypothesis that he was working on, to do these kinds of complex queries to just answer his question directly using SQL type of queries, right? So we really felt this is we need this Neo4j database. We need to be able to aggregate these really granular concepts in SNOMED into broader concepts that had larger numbers to find this pattern. So with that said, that's sort of the background and a sort of a preview of what we find. So let me show you the tool, and that's what really allowed us to bring Sandesh in to sort of do his deep thinking around his hypotheses of these different disease subtypes. So we had our data in a Neo4j graph database. We used a virtual machine at Duke, and then Tom Balmat developed a tool in R for us, and he used the Shiny package to do some of the interactivity pieces, and then there was also an interface called R Neo4j that then could within our R queries could then be translated into the Cypher queries, which is like an SQL kind of query language for the graph database. And then we had a Viz network package for generating some of the nodes and the edges from our queries. So here is a picture. I'm going to give you just a handful of screenshots as we sort of move through this tool that Tom designed. Bear with me here. We could actually have asked for a very specific concept, but I'm going to navigate the hierarchies a little. So we pulled in the Snomed Ct Hierarchy. So what you see here, you'll see body structure, clinical finding environment, right? So these are the high level Snomed Ct hierarchies. So we're just going to work our way towards tremor, which is a very particular neurological symptom that our expert was interested in. So we go first to clinical finding, and then we go to finding of movement. So I'm drilling down. And then we go to, where do we go? Involuntary movement finding, and then we go to tremor. And then we go ahead and select that concept, and then we need to refresh it. So this is a prototype. This is an early version of this tool. And then we've made that query on the SNOMED data. So we've selected the SNOMED concept that we want that and all the children for. And then the R script that Tom generated will query the UCD data in the Neo4j graph database and construct the nodes and edges that I'm going to show you in a minute. But first we needed to also sort of connect that with one of the variables in our data set, which was the Urea Cycle Disorder diagnosis. So this is a variable with the 8 different subtypes of Urea Cycle Disorders that I mentioned. And so then we get this is the visualization. I hope you can see it on the WebEx. It's a free form, there we go. It shows the different UCD diagnoses in gold. So there should be 8 gold dots here. And then the SNOMED codes in blue. And what's interesting is the one as the labels for these blue nodes here: resting tremor finding, chronic tremor finding, fine tremor. Those are all children. Those are part of the tremor concept that we selected. Then we can move this to a radial graph. So same information as in the first graph, but that one was kind of messy and hard to see. Now we've got all our gold nodes are the different disease subtypes of Urea Cycle Disorders on the right. On the left, we have the SNOMED codes that are in our data set here. And we can compare these a little bit more easily. I think I should mention here that there's 10 different types of tremor here, but on our display, there's 10 blue dots here. But in reality, if you looked at Snomed Ct, there are, I think, 41 or 42 children of tremor. So what we've done, if you will, is we've filtered the SNOMED, all those relationships that were in that database. We're just showing the ones that are relevant to our data. So that in and of itself was a huge breakthrough. This got us away from the hairball. Then you can see here that just sort of the nodes and edges the sizes and weights and rollover labels the sizes and weights are proportional to the number of records. And then you could hover over and see that 18 people had intentional tremor. 17 people have the urea cycle disorder subtype ALD, so it's a aldamine deficiency. And then you can also see that 12 participants had the ALD disorder and intentional tremor. All right, and these can all be adjusted, sort of the weight. There's all kinds of things that our Shiny package and the tool can allow us to configure. But now we want to look at just a couple. So that was distracting when we had all 8 disorders, right? So this is sort of driven by the expert now. It's starting to look at this and saying, okay, well, I'm not as worried about NAGs. OTC has a different sort of mechanism. I know about that. That's the most common of these conditions. So let's just look at ALD and ASD. So we have this functionality in the tool that we can then choose 2 nodes and click, alt-click, I guess, and select those. So then our visualization here now on the bottom right, you see ALD and ASD here. And so now it's just a little simpler. And this was the challenge with all of this, is just how to filter through and see what it is that we want to need and get rid of the noise, right? But there was never, always thought there was some magic button that would, noise filter or something, but it took us a long time to decide what's noise, and it's not anything you could do in sort of a build a generic tool at the start context, right? It's very much related to the question. So now, so we've got that information, but then our expert wanted to really look at, okay, well, what's the severity of disease, and how active is the disease in these two different subtypes, and what's the relationship between what's going on effectively in the liver in terms of the hyperammonemia, which we have a variable here called hyperammonemic events. And then, how do we connect that to what's going on with the neurological symptoms, and how does that vary for the 2 different conditions here? So we created there was a variable in the data set. And again, this comes from a very structured, very labor intensive study in terms of data collection. So very careful collection of a HA event would be a hospitalization event. There's all kinds of data associated with that. And we sort of came up with, or the study team had long ago had a variable. So does this person have a history of events or no events, right? So that's what we've got here. And then we wanted to then look at this even more closely in terms of combining an interaction variable. So here we start, we look more closely, we've got ASD with hyperammonemic events and without hyperammonemic events as brown nodes. Then we have patients with ALD and no hyperammonemic events, as well as patients with ALD and hyperammonemic events. And so each one of those is a distinct node, and each of those groups of patients have different phenotypes. They're showing different levels of different types of neurological findings recorded on the form. But again, to simplify, this is still a lot of nodes to look at. So Tom created in this tool this ability to make an interaction variable. So how can we combine the diagnosis and the HA events? Because that's what we're really interested in. So we create this interaction variable here. And let's see, actually, before I get to, I guess you could see, we're still talking about small numbers of patients here. So we've got just 8 patients with ALD and no hyperammonemic events. And this is starting with our data set of 733, which is a large data set for a rare disease. But, as you get in these questions, you start dealing with very small numbers. But anyway, as we start to build these interactions and look at these, I think might have brought the wrong slide. Oh, I do have the combination variables here. So anyway, this is really it. And I'm not doing justice to this, I think, because I'll emphasize my lesson learned in a minute, or I'll preview it, which is just there's such deep knowledge required in terms of the disease and the research questions, as well as the visualization, as well as understanding Snomed, that I'm not doing justice to this, but I will sort of repeat the insight that we gained. And this kind of came up on a call after a couple hours of looking at the data, that this was very insightful for our expert, who said that this visualization really showed that the patients with ALD and no hyperammonemic events are still showing resting tremors. So ALD and no hyperammonemic events, that's the 8 there. 7 of those are showing tremors, which is interesting and unexpected. So something's happening there that is not because of the urea cycle disorder, it's not because of hyperammonemia that's happening in the liver. There's something else happening, presumably in the neurons with these patients. And so this visualization really helps us understand that some patients are showing these neurological symptoms, not because of that accumulated ammonia in the body, but because of something else that's going on there, which can then help us decide, that's providing insight into that mechanism of disease and potential targets for treatment for those diseases. So I hope I did that. I hope I did that well. It took us a long time to get to this, and this is really the breakthrough slide. And you can see the point is there's just a ton of detail in here and nuance and small numbers. So this is not, we're not going to report this to Nature, but this is really supportive of a line of inquiry that Sandesh was working on at Baylor with different models of the disease. So as I sort of move us towards the discussion phase, I'll just say, obviously this is still a prototype in terms of the interface and the user design, and it's not a self-service tool at this point. We really needed to have Tom, who created the tool, really drive us on these, I guess I would call them just data exploration calls, where it was just a group of sort of the data analyst statistician that knew the data, the disease expert, Tom, a few people that were sort of so had some SNOMED knowledge like me in the middle, and we would just sort of play around on this, but we really needed Tom to drive it. I guess I'll just say the underlying data, of course, has issues. The coding was performed by different users. So these codes that we're looking at were put in there by humans at different sites. These are distributed studies. And so as I mentioned before, people use some are really careful in their coding and some aren't, either by choice or by sort of lack of training. This is, SNOMED, as everyone on this call knows, is really a complicated terminology. So there's some issues there, but I always thought that would just come out in the wash as we just start rolling things up into more general terms. I will say that we're limited in the data set. We only looked at one time point. So imagine if I actually put 10 years of data on these kids, even just the ability for them to have neurological symptoms or have a history of hyperammonemic events would give us more richness in this data, but just more to sift through. So we're not even ready to add a time point, time variables to this yet. And then, as I mentioned before, or I hope I alluded to before, there's still so many other data points that were collected in the studies. And quite frankly, the data that are of primary interest to the investigators in the study were collected not in this open-ended way with the SNOMED coding, but they were collected as structured variables. So we could really probably pull in a couple other variables to sort of reinforce this or sort of check the validity or make sure we're really comprehensive in the neurological symptoms and behavioral findings that we're looking at. So potentially what I'm showing out of this data that we looked at is incomplete. We could actually add some other variables there. And then I guess just sort of in general observational research in patients that enter research are a little bit different potentially than others with the condition. So as I'm wrapping up here, our challenges really was a lot of data. I know at the end we had really small sample sizes, but when we started 733 kids, 2 time points a year times 10 plus years, plus lots and lots of study variables, plus lots and lots of SNOMED codes, plus all the relationships of SNOMED, was really hard to just even design that. And then determining the correct level of precision to go at what point do we have enough cases or records to say that's the code we're interested in, is something I'd like to explore further. And I would love to hear if people have ideas on how to do that. Also, because of the polyhierarchical nature of SNOMED, if we looked at any given essential tremor, we'll have multiple parents. And so it's not clear which pathway. There's just lots and lots of potential pathways in this graph data-based model to sort of consider as a mode for sort of aggregating and querying things. And with that said, I'll mention that we also, as we were sort of using this tool to sort of look at the data and understand the data, there was a point where the counts didn't add up, that something was wrong and it didn't quite make sense in terms of, these numbers weren't adding up to what we thought should be the total. And as Tom went in there and investigated, we found, oh, there was an inactive Snomed concept in there. Well, the study went on for 10 years, and even the recruitment period was probably 4 or 5 years. So that's bound to happen, many times. And so, bringing in sort of the Snomed history in a graph format, would be a way around that, but there's definitely an opening for methods on how to deal with this. I think we just decided to happily take the explanation and ignore it, but that would be an issue of people doing this kind of thing in research. And that sort of leads me to this idea of needing counts and visualization. So we did a lot of back and forth between sort of looking at what's on the visualization, looking at the nodes and arcs and saying, what happens if we do this? Okay, now run that query again. Okay, let's eliminate those nodes. But then sometimes we wanted to see the actual counts. Tell me how many patients had NAGS deficiency and a seizure or something. And so then we wanted to go back to do that. So that's kind of one of the things that we'd love to potentially integrate into this tool. But I think our biggest challenge was really how to support these exploratory kinds of questions without an interface, but how do you build the interface when you're not quite sure what you want to find yet. So I'll just sort of end. I've got just 2 more slides, but the timeline just to highlight, these browsers were put in 2004 or something. That's when the Rare Disease Network started. The UCD Natural History Enrollment Study began, in 2005 or 2006. And then, participation was ongoing, recruitment was ongoing, then follow-up was ongoing for a number of years. And it's not really towards the end where you want to look at this kind of data in accumulation. And so, but we tinkered around with a lot of different approaches until we hit towards the end. And that's what I just want to highlight. When Tom came in and really helped build this tool, when we met the folks from University of Nebraska that just shared this Snomed in a graph form, and we had their paper to look at to sort of give us some insight, things moved. And then when we brought in the right expert with that visualization, this really moved fast. And I think we're moving toward a publication. We've generated something that he's put in one of his publications in one of his genetics journals. So, it's been really fruitful in terms of the science, just when we had the right teams and the right tools. And so we'd love to sort of improve this and make a self-service version. There's lots to do. We're not ready to share. It's not ready for prime time yet because we still need to refine it and build some guidance around it. But I'll just end with the importance of the multidisciplinary team. Like, this couldn't have been done. I think this is probably why it took so long, because we just needed we needed deep disease expertise, deep data expertise, deep technical and Snomed all in one room and with significant effort on this. But I think this could be a model for other diseases and challenge us to how to think about building interfaces for more exploratory kinds of questions using Snomed Ct. And then with that, again, I want to formally thank, I've mentioned Tom and Marcy, who really put so much work into getting this going, but certainly the whole Urea Cycle Disorders Consortium, particularly Bob McCarter and Sandesh Nagamani, were terrific, and Scott and Jay from Nebraska. We're just building on their great work. With that, I'll pause and I say a few minutes for questions or comments. So thanks for your attention.
Dr. Richesson, Rachel, oh my goodness, that was awesome. So I'm kind of, as a lot of people know, I'm impartial to data visualization. So seeing these node graphs of cohorts and variable interactions was just kind of like chef's kiss for me today. So that was great. Thank you.
Awesome.
Okay, so we do have some time for questions, and I do see one question in, so please go ahead and enter your questions into the Q&A box. Also, we do have a smaller group today, so please feel free to raise your hand, and I will then unmute you, and you can ask your question out loud. So, so yeah, prepare your questions, and we'll jump right in. So we have one question already. Is there a reason to choose Snomed Ct instead of Orpha codes, Orphanate, for rare disease coding in EHRs and data capture?
Yeah, but that's a great question, and I'm not sure I know the answer. I guess I would say, not necessarily. I'm not as familiar. I'm embarrassed. I'm not as familiar with Orphanate and their codes. I know there's semantics there, and I know there's a big connection to Snomed Ct. So I would think, the graph database approach and the tool, there'd be no reason that you couldn't use other codes and connect in. So this is not limited to Snomed per se, but this tool. I think we did Orpha codes might have been around when we were choosing Snomed for this and sort of designing the tool back at the start of the study when we chose Snomed as the representation. But I don't think it certainly wasn't on my radar at the time. And so that's sort of why we got Snomed Ct. I tried to decide what I thought had the most potential to sort of have people use for this coding, back in 2005. But I think if there's a link between Snomed Ct codes and Orpha codes, which I think there is, then those could be thrown in here. We could find ways to do that. So I think this doesn't negate using any other code system. And in fact, we have a lot of data coded in RxNorm that we haven't thrown in here, but I know Scott Campbell in his work at University of Nebraska also has RxNorm and in this graph database form and is using it. So I think Orpha codes, I would imagine, could easily be fed into this, and that this type of approach would work for them as well.
So I can also pipe in a little. So we actually do have a collaboration agreement with, or I should say INSERM, those who own and maintain Orphanate. And I'm not quite sure when your cohort was coded with the Snomed Ct terminology data. I missed that part on the timeline. However, we have incorporated, I want to say about 3,500 or maybe a little bit more Orphanate codes or Orphanate rare disease terminology terms into Snomed Ct. And we also have a Orphanate Snomed Ct equivalency map that's currently out in alpha release. And we are set to prepare to release the production, I think later this year, I want to say October time. So we do actually have a lot of the Orphanate codes already included in Snomed Ct. And we're looking at the next collaboration agreement with INSERM so we can incorporate more to have further alignment with Orphanate. So yeah, depending on when these particular cohort data that you have, Rachel, were encoded with Snomed Ct, those actually would have already been included, or you'd get actually even stronger, I guess, alignment with some new rare disease data. So.
Yeah, awesome. Thanks, Susie. Yeah, I think it went on for a long time. So it was as early as 2005 to probably.
Yeah, I wasn't quite sure of the timeline. So yeah. We have another question from Darren. Is the tool available for download?
Great, thanks. Not yet. We're still doing some work. So what I would say, if people find this interesting, please do reach out to me and let me know. What we're looking for at this point is it's just not quite ready. There's things that the way we're connecting to the graph database in terms of sort of the efficiency of those queries. I think there's a lot happening in the tool and the interface level that we could be more concise. I'm not virtually. There's some work to be done to make this useful, and we're not quite there yet. But I would love to have collaborators to sort of help us sort of think through these things and work to the next step. Our most immediate is to get something that's not really downloadable. It's because this is so configured to the data set, it's so tied to the data set. The whole query mechanism is sort of hard coded to the variables we had in the data set, that we're thinking about how to sort of make this useful for others, but really we're looking to, it just requires a lot of work to use it, and it's not quite the most efficient yet. So we want a little bit more time to sort of get it right and think about things. But if people are interested or have ideas for queries and particularly research questions around how to approach SNOMED, what are rules of thumb for using, some guidance on how to choose paths and that kind of thing, I would certainly encourage you to please reach out to me and just stay tuned. We're trying to move this forward.
Next question from Dr. Edward Chinham. Given your timeline, did you find the changes to the cipher query language added complexity to the analysis, a personal experience, or had it stabilized during your investigations, or perhaps you were insulated from this by the R language bindings?
Yeah, I think we were, I certainly was insulated from this, and I think we came in when perhaps cipher was a little more stable. Because, well, I don't know the last few years. I'll pause for a minute. If Scott or Tom want to jump in on this, maybe Scott, I will let him. Otherwise, I'll say I was insulated by it, and I think we just did this in the last, say, 2 years. So it wasn't a challenge for me personally.
So actually, Scott is asking, graph database tool and installing Snomed CT is available on Snomed site.
Yay. Okay. Oh, great. Okay, I didn't know that, Scott. And then Scott's got his work on GitHub too. If people are interested in this graph database representation, I think I probably assume people had more background than they did, but Scott Campbell has a wonderful paper in Journal of Biomedical Informatics on this. And if you did Snomed CT graph database, you'd find it. And then there's information to sort of get to some of what they've done and that they're sharing on GitHub.
Awesome. Thank you, Scott, for that info. And Mukesh says, I'll be interested in this tool as well. The presentation is really impressive. Great work. And yes.
Yeah. Thank you. Yeah, I knew this question would come up about sharing, and we're very much, we're doing this because we really are excited about advancing the use of data, in particular in rare diseases and these handful of data sets that have this kind of SNOMED coding. So, our spirit is definitely to move this forward. We just want to make sure that it's as done as we can make it. It's ready. So just stay tuned, and we're going to submit to Journal of Biomedical Informatics as well as sort of a response to Scott Campbell's paper. So hopefully you should see a more detailed version of this there soon.
Yeah, that'd be awesome. Yeah, and if you go ahead and
stop sharing
this on other domains and whatnot, please keep us updated. And yeah, we'd love to have you present again.
Terrific. Well, thank you so much for having me. This was really fun, and it's great to see some familiar faces there and to get good feedback. So I appreciate that. Definitely.
Thank you. And did another question just sneak in before? Okay. All right. And with that, I want to thank you, Dr. Richesson, for presenting for us this month. And I want to thank all of you who were able to take the hour to attend today. And we are still lining up someone for next month. So stay tuned. Again, you can sign up for the research reference group to get all the information on upcoming research related news. And otherwise, I will see you all next month. Thank you all. Bye.
Bye-bye. Thanks.